exactly what we've always talked about and what we will this time as well. Um, I love these. You know, we talk about a wide range of subjects because that is AIS. Right? Those are invasive species that is the nature of invasive species biology in the first place. Right? And there's a lot of disciplines involved, whether it's genetics, whether it's ecology, whether it's hydrology, whether it's engineering. I just like the multidisciplinary nature of it. Okay. We do have a website. It's right there. So if anybody misses anything on this, if you want to share it with somebody, I'm recording this now. We'll take this recording and we'll post it to the website. Actually, it's a YouTube link, but it works. We'll post it to the website so everybody can go back and scroll through it. If there's something you missed, or like I said, if you just wanted to share it. Okay. A little bit about funding, which we'll probably talk about towards the end. Previously funded by Fish and Wildlife Service for 3 years. Those 3 years are over, so we're currently operating without funding. So that's one of the reasons we're doing these online meetings. If everybody could please mute your microphone if you're not speaking, that would help a lot. I do this every time. So today's agenda is a good one. We have many things to talk about. Heidi McMaster is going to talk about Lower Colorado River Water Quality Monitoring Program. I'm going to talk far too much, but there's a few things I'd like to go over as well. One, opportunities that.
Hey, Dave, you keep going in and out.
I'm sorry. Is that better?
Dave, you keep, yeah.
Is that better? All right, we'll give it a shot. I don't know why.
Sounds better.
Okay, cool. So a lot of things to talk about today, different strategies, YY males to control nuisance fish populations in the Southwest. We've talked about that before, but today we're going to have Chad from the U of A talk about that in more detail. I do want to talk a little bit about cyanotoxins today. We usually talk about those a little bit, but we are going to pretty much go over what the EPA has advised for recreational ambient water quality criteria. We kind of always need to talk about gene drives because it's such a potent tool, right? So we're going to talk about that today. And then we're going to talk about the future of doing just these task forces, okay? So Heidi, did you want to go ahead and talk next? Do you want me to open it on my end, or did you want to go ahead and try it from yours? Heidi?
And it helps if I'm not on mute.
Never helped me.
We can do it from your end.
Okay, that's fine. Let me open it up for you here. Hang on. There we go. Okay, there we go. Can everybody see that?
All right. Well, for those that don't know me, I'm pretty sure everybody on the phone at least has heard my name. Yes. Can you hear me okay?
I can hear you fine, yes.
Yes. Okay. So for those that don't know me, my name is Heidi McMaster. I work for the Bureau of Reclamation in the Boulder City office. I am an environmental protection specialist, and for the last 3 years, I've been managing the water quality program for reclamation and been the Quagga Mussel coordinator. However, Jeff McPherson, who's also on the call, is new to us, and he's going to be taking point on all of the aquatic invasive species part. But I'm going to give kind of an overview, and I apologize, some of you have heard at least parts of this before, but at the North American Lake Management Society conference, I put together an overview of our program, and it was well received, so I'm going to deliver it again for those that may not have seen it. You can move forward. So I'm going to give a quick background, our program overview, and I'm going to talk specifically about Lake Mead, Lake Havasu, our contaminant monitoring program, which is our river sampling, and some additional concerns that we have. We have some additional sampling that we do, but it's not part of the Colorado system, the river system per se, so I'm going to leave those off for this talk. All right, go ahead. So as you can see from the map, this is our watershed, and we manage from below Lake Powell down to the Mexico border, and it covers almost all of Arizona, New Mexico, Nevada, Southern California, Southern Utah. Go ahead and scroll. So our primary driver for our program is the Colorado River Water Quality Improvement Program, which was brought about by the Salinity Control Act of 1974 and Mexico treaties. The Lake Mead Limnology Program and the Contaminant Monitoring Program is funded out of this work plan that we have, but we also have the Las Vegas Wash Monitoring Program and a Palo Verde Irrigation District, which is separate. We work with the irrigation district at the Palo Verde Irrigation Dam. Go ahead and move forward. The methods that we use and the different things that we collect for zooplankton, phytoplankton, our anions, cations, and TDS, and we use a water quality probe to collect for pH, conductivity, water temp, our percent saturation, and our DO measurements, with turbidity being a recent addition to our probe. Our plankton samples all come from BSA Environmental that John Beaver analyzes for us. And we've got over 10 years of plankton data from him on our reservoirs. Go ahead and move forward. We also collect for chlorophyll, phosphorus. We collect total and ortho, and nitrogen. And most of these are with a 0 to 5 meter composite using a pool hose, like putting your finger on a straw in your drink. These are all, well, up until 2 weeks ago, a week and a half ago, they were being sent to Alpha Analytical Labs in California. We just recently awarded to a new contractor for our nutrient analysis. Go ahead and move forward. These are examples of the equipment that we use. I believe we are the only agency in the Colorado Basin that uses Eureka probes. At least nobody that I've talked to. I've talked to a couple people that are considering using these probes, but we're the only ones that I know of that are actively using them. We have the Manta 2, the Sub 2 at the moment, but we just upgraded to a Bluetooth connection PDA just like a couple weeks ago. And we use a Wisconsin plankton net. Go ahead and move forward. For our river and our wash methods, we collect a slightly different set of parameters. We collect for total suspended solids, total dissolved solids, our selenium, perchlorate because of the issues coming from the Las Vegas Valley. And we also collect for contaminants of emerging concern, and that includes your endocrine disruptors like DEET, sucralose. We have like a full suite of 80 contaminants, I believe, or something like in the area of 80. And we also are collecting for algal toxins. We expanded our reservoir algal toxins to also include the river. And move forward. And I know most everybody has seen this to some degree, but this is our 1983 versus now current elevation and the difference that it's made. Go ahead and move forward. So this is what an aerial view of Lake Mead looks like, and this would be full pool, or at least close to. This is 1984. You can see there's no bathtub ring on the lake. Go ahead and move forward a slide. And we're going to see what 2016 it looked like and how much lower the lake is. We have a lot more land area. The water is being condensed down, and it's also condensing down our contaminants. So we're taking a little bit more closer look at the algae and the toxins that might be in the water because there's less dilution factor. Go ahead and move forward. So I have data spanning from 1990 up until the present. The depth sampled at Hoover Dam used to be around 200 meters in depth, and now we only get to about 110 meters, give or take 5 meters. We have approximately 40, there were 40 sample sites back in 1990, and now we're down to 19 sites that we can get to today. So it gives you an idea of how low the lake has gotten. And we sample quarterly in February, May, August, and November. And that has been consistent over, the last 28 years, been that way. You went backwards. Oh, never mind. You didn't. Okay, so we have 3 different basins that we sample. One day is Greg's Basin. Go ahead and click again. Another day is Virgin Basin, which is where the Virgin River and the Muddy River confluences come in. Greg's Basin is where the Colorado River confluence enters. And then Boulder Basin is another day that we also sample at the Hoover Dam, but that's where the Las Vegas Wash enters into Lake Mead. So we sample at each of the confluences and open sites on the reservoir. Okay, move forward. All right, so our Lake Havasu Quagga Mussel program is relatively new compared to Lake Mead. We're operating under the Wildlife Coordination Act for managing invasive species in the West. So there was no water sampling program occurring, at least on reclamation scale, until the quagga mussel infestation occurred in 2007. And then it became well, maybe we should find out what the water is like. Day late and a dollar short, but we're doing what we can. And this I got at Lake Havasu at sundown. And this is probably preaching to the choir for this meeting, but it's the land of blue-green water, located in Arizona and California, the usual stuff that I typically share with the audience that's not from the area. But anyway, this is the information that I usually share. Surface area is approximately 19,300 acres. And based off the calculations that I get from our Boulder Canyon operations office, the water retention rate is about 2 weeks versus in Lake Mead, it's about a year retention rate. And for those that aren't familiar with that, it's how long it takes for a drop of water to enter the system and then leave the system. So, and I would say that the CAP and the MWD straws in Lake Havasu have a lot to do with the retention. All right, go ahead and move forward. Here's a map of Lake Havasu, and unfortunately, I did get an updated map created, but I didn't get it in the presentation in time. But we sample 7 sites on Lake Havasu. At the very northern end is the inflow of the Colorado River. And then we sample our open sites, which would be in the Thalweg or the typical river mark. which I did recently find out is not completely accurate thanks to Doyle Wilson's bathymetry study that he just finished. So what we thought was our river is not quite the river. It's changed since the development of the reservoir. But we also collect one site up in the Bill Williams, and we thought it would be an interesting site to add to find out what's flowing in from the wildlife refuge. But what we're seeing in the data, which I'm not going to present any raw data today, is that it's a slightly different planktonic community than the rest of the lake. So with some further analysis and some more data sets, that'll be an interesting comparison to do between Lake Havasu proper and the Bill Williams Refuge. Go ahead and move forward. So I'll talk a little bit about our algae problem. In 2015, in our March trip to Lake Havasu, there was a huge algae mass, just open masses on top of the lake. So we started collecting for algal toxins, starting with just microcystin, and then about 6 months later, we expanded that to include different variations of microcystin, as well as euglenophysin and some other additional algal toxins. But we got these pictures in May on Lake Havasu, and it was definitely a pea soup atmosphere going on there. So go ahead and move forward. Our contaminant monitoring program, which I had mentioned, is our river sampling. It starts from Willow Beach and goes to the northerly international boundary with Mexico. There's 14 sites, and we sample twice a year. And it's supposed to be indicative of high flows and low flows of the river. But we sample usually the first week of August and the first week of December. And it's just for our purposes of scheduling, we've kept it the same every year for probably 15 years. All right, move forward. And go ahead and click. There's some add-ons here, yeah. So the first day is from Willow Beach at site CR 330 down to Lake Havasu, which CR 210 is Rotary Park, for those that are familiar with it. Day 2, we leave Havasu the next day, and we start at the Parker Strip, and we move down to Yuma. And some of the sites are pretty remote. Some of them we're going through access roads from the irrigation districts. So it's definitely an interesting trip, especially after a rain. Gotta tell you that. All right, go ahead and move forward. So that's our general overview. Because what we do, we have a lot of different programs, projects for sampling analysis, and we're slowly working on analyzing our data to be able to present more detailed information. I had previously presented some Lake Havasu data, but now that's out of date. And when Janet Kirsch retired, it left a kind of a gap in our analysis. So we're slowly getting that back together, thanks with Jeff McPherson coming on board. So these are some of our references that I've used to work with our data and our projects. And go ahead and move forward one more. And our partners that we work with on a regular basis, all of our labs, and Alpha, I have to update. Eurofins, for anybody that wants to know how we get our contaminants of emerging concern, we use Eurofins Analytical, and they do a good job of providing us the data. And if anybody asks, it's a very expensive analysis, just throwing that out there. But I'd be happy if anybody else wants to jump on and use them, I can put them in touch. And one more slide, I think, and then that'll be it. Okay, so there's one more. So if anybody gets a chance to go on an aerial overview, we did a flyover at Lake Mead with the Park Service to look for algae blooms, and I managed to get this picture of the Colorado River coming into Lake Mead, and it was definitely a really interesting and cool trip to be able to do. So if anybody gets that opportunity to do in the future, I strongly recommend it. And now there's one more slide that just says, does anybody have any questions?
Any questions for Heidi? All right. Well, thank you so much, Heidi. I appreciate it. I have a question. You were.
Yeah, no problem.
Do you coordinate with other state agencies that are out on the river or on the lakes sampling for their programs too?
Yes, I actually just recently was coordinating with Arizona Department of Environmental Quality. They're sampling almost every other month that we're not on Lake Havasu. So we're working on our schedules to coordinate that. On Lake Mead, there's every agency under the sun sampling, and we coordinate with the Lake Mead task force up there, which a lot of the people on this call are also on that one. And then also for Havasu, I go out, let's see, our volunteers that go out with us include Chemehuevi, the Colorado River Indian tribes, and then Doyle Wilson from the city of Lake Havasu. We've also coordinated with BLM and talked to them about any concerns from the locals and things that we need to be worrying about. And we participate with Arizona Game and Fish on the Havasu coordination calls for the quagga mussel. But I don't know if that answers your question, but we've been trying to build up more of those relationships and working with the other agencies on the river.
No, that absolutely does answer my question. It's always easier to have help doing that kind of stuff, right? So,
oh yeah.
Yeah, I mean it's a big job, you know.
Oh, it is definitely a big job. You have another question?
No, I'm done. Thank you so much, Heidi. One thing I did do was introductions, and I know, like I said, this is like herding cats. But is there any way to go around the room and give brief introductions for everybody? So whoever wants to start can please do that.
How about you start with agencies? If you list off the agencies, that might help coordinate it.
That was a good idea. So can we start off with the agencies then? Whoever wants to be the
Hey, David, this is Barack Shamai, Fish and Wildlife Service out of Albuquerque, Region 2.
Awesome. Thank you.
I am the Aquatic Invasive Species Coordinator for Region 2, which includes Arizona, New Mexico, Texas, Oklahoma. Nice talk, Heidi. Thank you.
Thanks, Barack.
All right. Who's next?
Hey, this is Chad Teal and Melanie Culver. We're at U of A School of Natural Resources.
Thanks.
In the environment.
Erin Rainey with the Arizona Game and Fish Department. I'm the AIS coordinator.
Thank you, Erin.
Kate Stegler, Arizona Game and Fish, AIS specialist.
That's BLM, Zachary Soliday, fisheries biologist.
Ford Monty, wildlife biologist.
All right, Master.
Doyle Wilson, Kansas City.
Bethany Durango, Arizona Refuges ISST lead.
Rick Gay, Arizona Department of Environmental Quality and Monitoring Assessment Unit Manager.
Great. Thanks, Rick.
Carol Evans, Bureau of Reclamation, Phoenix Area Office.
Nick Heatwolf, Yuma Area Office, Bureau of Reclamation.
Hi, Nick.
Nelson Summerday with USDA APHIS.
Kurt Deucer, National Park Service, Lake Mead Interregional Exotic Plant Management Team.
Awesome. Thanks, Kurt. All right.
Colleen Allen, Glen Canyon National Recreation Area, National Park Service, and Kristen Lewis, Lab Technician.
Awesome.
Brian Moorhead, Salt River Project.
Sorry, Brian. Can you repeat that?
Brian Moorhead from Salt River Project in Phoenix.
Thank you, Brian. I cut you off there. This is Laura McGill from the Nevada Department of Wildlife. I am the AIS coordinator.
Awesome. Thanks, Laura. All right.
From the California Department of Fish and... Oh, go ahead, Kara, sorry.
Kara from California State Parks, Division of Boating and Waterways, in the supervisor of the Quagga Zebra Mussel Infestation Prevention Grant Program.
Deborah Herndon, Nevada Department of Wildlife, fisheries biologist.
And Dominique Norton, California Department of Fish and Wildlife. I work in our Quagga Zebra Mussel program.
This is Ian Ralston, aquatic invasive species biologist out of the Blythe Field Office.
This is Bill Stewart with the Bureau of Reclamation out of the Phoenix area office.
All right.
Melanie Gal with USDA out of Yuma.
All right. Is that everybody? Thank you so much for taking the time, first of all, because I did want to have that on the recorded version of that. And I just like knowing who everybody is we're presenting to. We've always had a diverse population of varied interests. So it's one of the things I really like about this task force. And I'm going to do a little shameless plug. But you know, kind of going hand in hand with what Heidi was just talking about. I mean, there's a lot of work monitoring the environment. There's a lot of work in the process of the ecology and finding out the science behind invasive species in general. So something I've been working on is this Center for Student Agency Opportunities in Environmental Sciences. I mean, most of us graduated in an environmental science degree of some kind, or wildlife and fisheries, or hydrology, or something close to it. And for the most part, you know, funding opportunities for those students are not as great as they are for other students. Engineering sciences, certainly, you know, the medical field, the health industry. So for environmental sciences, funding opportunities exist. Some internships exist, some assistantships exist, but they're far fewer than they actually are for other disciplines. Okay, go forward. One of the things I did want to talk about too is, you know, helping government agencies and private industry is part of our mandate as a land-grant university. It's our job to actually help and provide answers for these types of things, including AIS. Right? Arizona was still a territory when the U of A was established as a land-grant university in 1885. The nice thing about land-grant universities is that's exactly what we do. We're established to specialize in applied sciences, whether originally our mandate was to help agriculture, but also science in general. But for the most part, they were highly applied sciences. They were applied sciences to help solve specific real-world problems. Okay. And yes, my computer's taking a while to forward slides. I apologize. Come on. Anytime today would be good. There have been some obvious changes since the original land-grant universities were established. You know, we have societal shifts, we have demographic changes, we have certainly enhanced technologies that we can use, right? And we've kind of come to realize that it's not just our state that's important, but it's other states surrounding us, it's it's the globe that is important. If we think about something like quagga mussel, which came from well outside the United States, and a lot of these invasive species will come from other areas. So education and research in the area of natural resources has always been a priority for every land grant institution. And yeah, if we start looking at funding, this is for fiscal year 2019. Some of these are projected, but I think that most of these are well established. It's not going up. A lot of them have managed to stay the same. Fish and Wildlife Service, for the most part, funding for natural resource management agencies is on the decline, right? And there's hiring freezes for some places, for some agencies, not all. And it makes it difficult to actually get work done, to fulfill the mandate of that particular agency. So times are tight. Forward would be good. There we go. This just shows, proposed changes from 2017 spending to 2019 spending, right? So we look at, NIH has a slight bump, so does NASA. National Science Foundation is about the same. But things like EPA taking a definite hit, and DOE energy programs, even a greater hit. So, like I was saying, times are tight out there, so we have to kind of make do with what we have. So the premise of the center is, it's a dual opportunity for students, graduate or undergraduate, and research management agencies to get work done, right? In the way of applied science in a world of shrinking budgets, which is exactly the case now. It also gives students invaluable experience. I don't know about you guys, but when I was a student, I was willing to do just about anything. I just wanted to be out in the field, or I wanted to work on anything that was more applied to my particular discipline than taking tests and reading books, even though that is important. Don't get me wrong. I teach a couple classes. I shouldn't be saying that. But at the same time, we want field experience, right? And that field experience can be used, I know mine was, as part of a thesis, dissertation, or potentially for university credit. And yeah, like I said, there are internships out there, right? And there's just not enough of them. I teach limnology, I teach algae, I teach aquatic plants, I teach ecotoxicology, and I'm constantly barraged with students asking me if there's anything that they can work on, right? Sometimes for pay, sometimes for not. So I know that the need among students is definitely out there. Right? And it's not just, field work. It's also laboratory work. It's data entry. It's running statistics. The thing about a program like this is, we all kind of have projects and we get funding as principal investigators, and we usually hire students, varying skill levels, whether an undergraduate, a graduate student, or all the way up to a postdoc. But they usually take a secondary role. Yes, we oversee what they do to the nth degree. In this, projects would be still overseen by U of A faculty, but at the same time, it would be the students that would be doing most of the work, being well monitored, but still doing most of the work. It also gained experience geared toward a natural resource agency's particular mandates. I don't know if a lot of students even realize Fish and Wildlife Service even is, or USGS, or BLM, or Bureau of Reclamation. There are some classes that teach them that, but before they take those classes, they really have no idea. As far as travel goes, we all work on projects all over the place. I tell people historically I'm an aquatic ecologist in Tucson, Arizona, and they look at me like I'm crazy, more so than normal. But, we do have projects elsewhere. Right? Cooperative extension offices, as part of a land grant university, we have extension offices in every county. That's our mandate, right? So it's not anything where they have to do something on campus. We could send students in the field wherever a problem happened to occur. Facilities, gear, and infrastructure. We have a lot of that here. I know I have a lot of lab equipment. I have a lot of field gear. We have boats. We have stream sampling. We have whatever you need. We already have that. And a lot of times it's honestly, for a lot of my stuff at least, it's not used as much as it should be used. I work in a aquatic toxicology lab, and I know it is a very underutilized facility. So instead of having to go out and purchase new things, we have facilities, gear, and infrastructure kind of under one roof, in addition to student help. And there is a misnomer out there that academia can't consult, and well, we actually can. In fact, that's a large chunk of what we do. Universities can perform all the services an environmental consulting firm can. Okay? We can subcontract with them. We often do. We can pair with them. We can have them as subcontractors. I mean, all of that is a possibility. Okay? And the nice thing about a program like this is students can be provided with all the necessary agency mandated training. We have classes here already geared towards that, whether it's HAZWOP or whatever you need, right? A lot of students will already have that training. So the student involvement can be a range of almost anything. I mean, the projects and the tasks don't have to be all that large. Like I said, it could be anywhere from data entry to running statistics to, I don't know, GIS to whatever you have. But we would go to the trouble of actually matching the student with any particular task. And we could, we have a large group of students to choose from. It could be undergraduates, it could be master's level, PhD, or postdoc. Obviously, as we go up to the master's and the graduate level students, the problems would be more complex. Right? So the discipline and the experiments would be matched with any given project. Student benefits. Well, there's potential funding. It doesn't have to necessarily be funding, but the potential for funding is there. Upward positioning for future employment. I know USGS has their SCEP program. It's a great program where students, particularly grad students, will work for USGS, and then they're first, well, they're not first in line, but they've moved up in the line towards potential employees in the future. Okay? So that's a big impetus for students to join something like this. There's also resume building, skill building matched to a specific agency. Right? They would get firsthand experience of a particular agency's mandates. Right? And the big deal is just experience in general. Agency benefits, yeah, you can probably save money compared to hiring a consultant firm or additional employees, even if that's possible due to potential hiring freezes. We could actually get work done under the conditions of shrinking budgets. We can help with projects, we can do projects, or anything in between. There is a certain amount of credibility by having a university involved sometimes. You'd also, in the long run, get potential future employees with a head start on that agency's specific mandates and operational goals. Okay. So I know this doesn't have anything to do with AIS, but I still wanted to mention it as an opportunity because it would be a great lesson in mutualism, right? Agencies would benefit, students would benefit, and such a program like this kind of fits exactly into the mandate of a land-grant university. That's why land-grant universities were formed in the first place. Okay? This is our goal, is to help agencies. All right. Does anybody have any questions? No.
Hey, David, this is Heidi.
Hi.
Not so much a question, but this your presentation is very well. Well, I've had this discussion with other agencies, state and federal, about how the we're out of the environmental program and trying to hire new students, fresh into the scene, they're missing that field experience, that hands-on. And it would be a real benefit to get, some of these students, whether bachelor's or master's level students, out to help out and get that hands-on experience, so that when they are looking for the job, even if they're just out there volunteering for various parts, it would help out be able to get that hands-on requirement to be able to be hired. And we take out our interns when they're brought on, but they're not always there. They're usually set for someone else, but we will take them out. But if we can get assistance with our projects, I think I know volunteering is really easy for us to work with, but we can help get that experience up for students.
Absolutely. I mean, that's kind of what this is geared towards, kind of be that bridge between being an intern at an agency versus having an academic career, right? Because there's a big gap in there. Even when I graduated, well, I had been working for agencies for a while, but when I graduated, I had no clue what agencies really needed, and what field work was really like. So there is that, and that is kind of the exact goal of this. Now, I know that there are citizen volunteer groups out there, and those are great, and I'm not knocking them at all. I think that they perform very vital functions. But students, they have a background in this already. What they're lacking is exactly what you just mentioned, is that field experience to go out and say, okay, here's what I've learned in class, or here's what I've learned in books, versus what I'm actually seeing out in the field. And sometimes those are a world apart. So thank you, Heidi. Any other questions? Okay, cool. So next we have, Chad. Chad Teal. Chad, did you want me to go ahead and run it from this end, or did you want to try and take it over, or whatever you wanted to do?
Yeah.
I'm sorry, what was that?
Yeah.
Oh yeah, you can go ahead and click through the slides for me if you don't mind.
Yeah, sure, of course. So now we have Chad Teal.
Can you hear us all right?
Yes, I can hear you.
Okay, great. Good afternoon, everybody. My name is Chad Teal. Currently a PhD student with the University of Arizona School of Natural Resources, focusing on fisheries conservation management. Started my PhD this past January, and some of this presentation might be a review for a lot of you guys, but I'm going to go ahead and fill you in with what I've been up to for the last few months, and how we can take this idea of using YY males to control nuisance fish populations affecting the Southwest. So go ahead and click the next one for me.
I'm clicking, it's just taking its time. Sorry.
That's all right. There
we go.
Right, so you guys know the story. We have lots of non-native fish. A lot of these fish are providing huge monetary benefits to Arizona, New Mexico, $1 million sport fish industry provided by these non-natives. Sometimes they can become a nuisance though. For instance, the red shiner, the western mosquito fish, these guys are competing with some of the native cyprinids, and even some of the sport fish, when they get out of areas where they're not being managed, can cost millions of dollars in mitigating their effects. So the question and the idea behind the project is, how can we be very specifically target species that are in nuisance populations in areas around the Gila River Basin. So you can go to the next one.
It is, I think. Come on. Ah.
So pretty much, what's that? Oh, there we go. There it is. Yeah, so pretty much ever since Watson and Crick, people have been trying to think of ways to use pest genomes in order against themselves. So using autocidal pest control. And combining that idea with traditional aquaculture practices, such as developing a monosex population, which has been something that's been going on for decades, using hormones in order to develop monosex populations of commercially viable aquaculture species. So combining these two ideas, go ahead and click to the next one for me, David. I'll just tell you to click like when I'm pretty much almost finished with what I'm saying, or halfway through what I'm saying.
Thank you.
Get through this a little quicker.
It's going. Come on. There we go.
All right. Why don't you go ahead and click to the next one? But what I'm going to say is, so the idea was modeled back in 2006 by Gutierrez and team, and they saw that we could use YY fish, so fish with 2 Y chromosomes, as a means of population control, and that this would be an effective alternative to using something like gene drive or CRISPR. And it's a little bit, there's some studies that show that people are a little bit more receptive to using this form of population control over gene drive and CRISPR, because you're not actually introducing any new genes or any deleterious alleles or code into that genome. So if there's any issue with escapements of these YY fish, we don't have to be too concerned about it having big ramifications farther down the line. So so Gutierrez and team modeled that it works, and then Dan Schill, he was with Idaho Fish and Game. He just retired recently, but he wanted to take this theoretical biology and make it a reality. And so he developed YY male brook trout. And so the idea is that you're going to produce all these YY males, you're going to release them into a nuisance population, and if you put enough of them into a system, you're going to skew the sex ratio enough to the point, or you're going to skew the sex ratio towards male enough that it becomes an ineffective population. So this is just a table from one of the papers that they modeled specifically for YY male brook trout releases, and it's pretty intuitive. So the higher your suppression rate by using just traditional removal efforts, the less your stocking rate needs to be. So that just makes sense. But you have to stock at a pretty high density to begin with, but you can get it down to less than 10 years to completely extirpate a nuisance population using YY fish. Specifically for this study, it was brook trout. But this has been modeled with western mosquito fish. This has been modeled with carp, and so it's looking like a consistent theme in the models that this is something that's feasible. Probably best to be done in conjunction with traditional removal efforts, but it should make it more cost effective to use these YY males in conjunction with using pesticide treatment or rotenone treatments and fish barriers, things like that. So it's just another tool for fish managers to have. You can go ahead. Yeah, there we go. Perfect. So my project is looking specifically at the Gila River Basin, and it's sponsored by U.S. Bureau of Reclamation with Bill Stewart. So the 2 species that we're looking at that are a big issue in this river system, which is a tributary of the Colorado River, is the red shiner, which is a very prolific breeder. It can spawn many times within a year. It competes with many of the native small-bodied cyprinids, such as spike dace and loach minnow, and it's yeah, like I said, they just pump out babies like crazy. So that's our short-lived species we're looking at. The more long-lived species we have in the lab right now is the green sunfish, which is a voracious predator. Can gobble up it'll gobble up anything it put its mouth around, and it also is very aggressive. So it tends to compete with more, I guess, favorable sport fish such as bluegill and largemouth bass. So they can be an issue even in areas that are managed for sport fisheries. Go to the next one for me. So this is kind of where we're starting at. Over the last 6 months, we have a lab here at the University of Arizona that we completely renovated for the sole purpose of producing YY male red shiner and green sunfish. So Heidi Blasius with Bureau of Land Management put me on a spot in the Gila River where we collected about 150 of these guys to act as the red shiner to act as our broodstock. And then Nathan Berg with Arizona Fish and Game Department has taken me out a few times electrofishing in Parker Canyon Lake for the green sunfish broodstock. And those are the fish we have in the lab right now. You can go to the next one for me. And so where we're starting is just figuring out how to spawn these guys. So on the left we have the larger male green sunfish is trying to get this female to spawn, and then in an artificial nest that we have in some of the tanks. Then on the right we have these spawning stacks. They're red shiner or crevice spawners. So we have this set up in our lab, and we're getting the red shiner to spawn very dependent, like they spawn every 5 days. Green sunfish have been a little bit more tricky. We're building some new systems to get them to more routinely spawn, but we only had one breeding pair spawn so far. So yeah, this is where we're starting, is just using our broodstock and figuring out how to breed them. Go ahead and go to the next one. And this is the method for developing YY fish. So you guys might have seen this already if you've heard about YY fish production, but it's pretty simple. You take your breeding pair and the progeny from that pair, you split in half. 50% of them get a hormone treatment, typically estradiol or estrone, some sort of estrogen hormone. The other half of the spawn doesn't get any treatment. So what you're gonna hopefully do is hit these larvae with this hormone during the labile period of their development. So they won't go back in their sex if you hit them at the right time of their development. So you'll have a phenotypically female or male fish that's producing eggs. Then you'll spawn that female with an XY chromosome and with a traditional wild type male. So then when you spawn those two, when you breed those two together, go down to another generation, you have theoretically 25% of those spawn are going to be YY males, and you can sex reverse them so that they'll be egg producing YY individuals, and then you breed them with regular, well, not regular, but male YYs, and that is the offspring of the female YY with the male YY is what you release into a nuisance population. So it's got a added sort of warm and fuzzy aspect to it, because you're not releasing any hormone treated fish into the system. So that tends to settle people's ease, and there's no way that they're going to sex revert or go back to a female YY if they're not being exposed to the estrogen. That is those Trojan sex carriers at the bottom. Let me see, anything else I want to talk about with this? And the same can be done if you have a fish with a ZZZW sex determination. So that being that the female is heterozygous, so the female is the ZW. The only thing is that you would be releasing a hormone treated ZZ female into the system. So you're actually taking the fish that was sex reversed and putting it into the water. Yeah, go ahead and go to the next one. Right, so like I said, we've been spawning these fish out and then going through larval rearing of the different species. So we've gotten the recipe down for producing lots of baby red shiner. They've been very straightforward with producing the green sunfish. We're working on a couple different ways to rear them. So we should have some bigger broods and bigger spawns coming from them in the next throughout this semester through fall. So go to the next one for me. So a huge aspect of this project is developing sex identification markers and determining hopefully that there is some sort of genetic sex difference between the males and the females of our target species. So the way that we're doing that with Dr. Melanie Culver's help is using RAD sequencing, and one of her students, Alex Irwin, has been helping me tremendously in using RAD sequencing to look at differences in the genomes between the males and the females. So since we don't have a fully sequenced genome, or even a large part of the genome of green sunfish and red shiner coded, we have to use reference genomes of I'm using a yellow croaker for the green sunfish and a goldfish for the red shiner in order to determine what restriction enzymes to use and what techniques will be best for developing the RAD library that will be used to build the sex ID markers. So right now we've extracted 18 green sunfish, or DNA from 18 green sunfish and DNA from 18 red shiner, and those will be used, those individuals will be used to develop the sex ID markers. You can go to the next one. So yeah, that's where I'm at right now. Like I said, we've developed spawning protocols for red shiner, green sunfish, and we've got the basic aquaculture aspects kind of under our belts, and now we're moving into more of the genetic side of things. I'll be starting the actual sex reversal treatment in next month. So I've been making batches of different estradiol treated feeds. And so we'll have a couple different treatments just to make sure that we actually have a successful sex reversal of the fish that we're treating. If you give them too much estrogen, you can cause sterilization. If you give them too little estrogen, you'll have intersex fish. So we have to play around with a couple different recipes on how long we give the hormone treatment and at what concentration. And I also want to start looking more into the modeling and seeing how we can get some of the different variables on our species and put them into a rough model to see, what numbers do we need in order to, what's the stocking rate, what's the percentage of the wild population that we need to reach in order to have a real effect and drop, if not extirpate, this whole population from a specific area. I think that's it. The next one just a slide with acknowledgements. And I just want to thank you guys for giving me the time to chat with you about my project. If you have any questions, I'd be happy to answer them.
Awesome. That's great, Chad. Any questions for Chad? No? No. I think that's fascinating. We've kind of talked about YY males before, and I think it's, for a lot of these problems with invasive species, it's going to depend upon some type of genetic manipulation. And like Chad was saying, there's far less of a creep factor involved with, using YY males than there are, gene drives or anything else. We're going to talk about gene drives in a few. But yeah, having alternatives to gene drives are super important. One of the things I wanted to talk about today. Thank you.
Hello, I got a question.
Yes.
Kurt Deucer, you hear me?
I can hear you, yes.
David?
Yes.
Yeah, excellent.
Yeah. Are there some good examples of this being applied, you know, in the environment and other animals and all being successful? And
Yes. So this kind of idea has been applied in mosquitoes, and mosquitoes. And I think I neglected to mention that Dan Schill with the brook trout, he's actually put these YY males in some reaches of different streams and in ponds, and has seen that they are displaying, a level of fitness which is going to allow them to actually be successful, and that they were actually spawning with the wild type females. So he has some real world field-based results coming in from these YY male fish. But yeah, also it's like I was saying, the autocidal pest control has been looked at with mosquitoes and developing sterile males to compete with, fertile males.
right, relate all the disease transmissions from mosquitoes and such. Yeah.
Mm-hmm.
All right. Any other questions for Chad? All right, thank you, Chad. I appreciate it.
Yeah, thank you.
Absolutely. And if anybody has any questions later, feel free to email me and we can pass it along. One thing I did want to talk about today, which actually isn't my presentation, but I wanted to go through it. There was a recent EPA meeting talking about revised recreational ambient water quality criteria and or swimming advisories for cyanotoxins. And it's worth going through, especially in regards to what we know about cyanobacteria and cyanotoxins now. Algae in general are among the patchiest, environmentally patchiest populations out there. Okay? They're very hard to determine a why as to why they're there and where they exist, why they exist in a certain region. So, they have this science-based approach, looking at actually creating policy, which is great. It's very difficult to do. So just a little bit about their outline, background information, and revising it in response to public comment, which is always good. Nobody likes to see beaches closed, right? Nobody likes to see lakes closed. So public comment is always important. And then some of the implementation tools, okay? And then gave some background information on water quality advisories and swimming advisories. So in 2015, EPA published the health effects support documents that describe the human health effects from exposure to the cyanotoxins microcystin, should probably be plural, cylindrospermopsin, and anatoxin A. Now these are target toxins, and with good reason. They're fairly ubiquitous in the environment. Anatoxin A has a very short half-life. It's degraded by sunlight and hard water or total alkalinity, which we're not lacking in Arizona. So in Arizona, at least in the arid Southwest, anatoxin A or anatoxin AS, which is a variant of it, really we wouldn't expect much of a problem. And actually just trying to sample for anatoxin A in the environment is difficult. Samples should be immediately frozen, Dewar's flasks, that kind of stuff. So they mostly are concerned with microcystins and cylindrospermopsin. But like Heidi was talking about earlier, there are a lot of different types of algal toxins in the environment. Not all of them are cyanotoxins. Euglena produces a unique toxin. There's saxitoxins produced by dinoflagellates, which we do see inland, rather than just, brown on red tides. But they wanted to focus just on these because rightfully so, like I said, they are the most ubiquitous. Right? So 2 drinking water health advisories of 2015 were developed for microcystin and cylindrospermopsin. Upon publication, EPA received questions about effects from exposure during swimming and fish consumption. Now, cyanotoxins, at least these 2, not all of them do, but microcystin and cylindrospermopsin does bioaccumulate. So sure, there are real world concerns about what's in these fish that are being caught for consumption. It does not biomagnify, okay? But you can probably go out to any of the lakes along the Colorado River, and during certain times of the year, you can probably find either microcystin or cylindrous spermopsin in their flesh, okay? Most readily consumed part of fish. So they had this development and stakeholder engagement, initiated development of values that reflect the latest science to protect the primary contact in recreational use. And I'm not going to lie, the science behind, even why some of cyanotoxins produce toxins is still muddy. I have looked at one cell of cylindrospermopsis, which produces cylindrospermopsin, and that individual cell, produces toxins sometimes and doesn't other times. So it's very difficult to be able to predict with any degree of certainty the amount and concentration of toxins that are going to be in the water. Okay. Anyway, they arrived at these recommendations for values that states can either use the 304A recreational criteria or as swimming advisories, or both. Okay. So most of this is for notification purposes. Obviously, there's a public health concern. It uses peer-reviewed science to develop recommended values for microcystins and cylindrospermopsin to protect the recreational use. Recreational use is a big deal. Okay, we're going to get into this a little bit later, but they also talk about drinking water quality. Most of these toxins, even non-cyanotoxins, are readily removed via even conventional treatment. So, yeah, it's a concern. It's not as big of a concern, rightfully so, from a public health standpoint as are recreationists and recreational uses. Okay. So they evaluated the latest science describing the health effects from exposure to cyanobacterial cells. All right. So that's kind of a nebulous relationship. Because like I was saying earlier, not all cyanobacterial cells produce toxins. The vast majority of them don't. But there is a correlation, between the number of cells in the water and the amount of toxin that's actually being produced. It's not a great correlation, but certainly there is. In other words, for cyanotoxin to be there in the first place, you have to have cyanobacteria. Right? So they came up with this conceptual model, like I said, based on cyanobacterial cells. I would like to see more research being done on cyanobacterial cells and why they actually produce toxin. There has been some work along those lines, but before we come up with the criteria of cyanobacterial cells being negatively implicated in a public health realm, we probably want to understand more of why they produce toxins, and that's not as well established yet. Okay. So they came up with, the sources, obviously, but the exposure routes. There is recent research talking about cyanobacterial cells and inhalation or aerosolization, and that does occur, especially if you happen to be on a beach or in an area where, there might be some surf or at least some type of tide coming in. Cyanobacterial toxins can become aerosolized and you can breathe them in. Obviously, the primary exposure route is oral. Okay. And these are hepatotoxins. Okay. Dermal, swimmer's itch, that type of thing can happen with or without cyanotoxins. In fact, dermal happens primarily without cyanotoxins. That's more of a green algae type of thing. But getting back into it, these are hepatotoxins. They'll affect liver and kidney. There are some reproductive effects, especially in aquatic organisms, developmental effects, cancer, carcinogenic. It's very low on the totem pole, but especially things like liver and kidney or hepatotoxins. Okay. And like I was saying, recreators are exposed almost, I can't say solely, but for the most part by the oral route. Okay. And it's just through incidental ingestion of water while swimming. Okay. And it's actually greater in kids than it is in adults. It's especially large in things like domestic wildlife, especially things like dogs, right? I have a dog. He's not, if that water is green and full of microcystin or cylindrospermopsis, he's going in the water regardless because he doesn't really understand. So there is that. Dermal inhalation pathways can be important to consider for recreational exposure to cyanobacterial cells. Yes, they can be, but like I was saying earlier, by far and large, the primary route of exposure is going to be oral. That's going to cause the most concern. What's interesting is that children, I don't mean to relate children to my dog, but children share a disproportionate share of the incidents surrounding HAB-associated outbreaks. Green water is cool if you're a kid, right? You want it on you. You want to swim in it. So 66% of the outbreaks in 2009 to 2010 were among children less than 19 years of age. 35% were less than 9 years, and 80% of all confirmed illness reports due to freshwater cyanotoxin exposure involve children. It's a significant finding. Okay. Children also are more prone. They're at a critical life stage. They're, not only the greater potential exposure, but they're more prone to the effects of cyanotoxins. They also have a much smaller body size, obviously, so the concentrations wouldn't have to be as great in order to cause harm. Okay. So EPA came with its draft recreational water quality swimwear advisory recommendations of 4 micrograms per liter for microcystins and 8 micrograms per liter for cylindrospermopsin. That is not to be exceeded on any day, which is great, but usually nobody's out there sampling the water, especially on beaches, for cyanotoxins every day. Recreational water criteria not to be exceeded more than 10% of days per recreational season up to 1 calendar year. All right. Where they came up with the 4 micrograms per liter and the 8 micrograms per liter, I think was more of a statistical thing rather than it was a strict physiological thing. Right, and here we get back into this again. Documents describe available data on effects related to total cyanobacterial cell exposure. Right? Which is, like I said, is fine. There is a correlation there, it's just not a good one. Elevated cell densities associated with inflammatory health endpoints. There is a linkage between exposure to total cyanobacteria and health effects, obviously, because you have to have cyanobacteria there in the first place in order to have cyanobacterial or cyanotoxicity. Available data were insufficient to suggest a nationally applicable value for the reasons I was talking about earlier. It's really hard to determine in any HAB which cells actually might be producing toxins versus those that actually aren't. So they have this significant density range of 5,000 to 100,000 total cyanobacterial cells. That's a pretty big range, right? We find 5,000 cyanobacterial cells even in lakes and reservoirs that are kind of ultra oligotrophic. So, yeah. So they posted this for public comment December 2016 to March 2017. They got comments from 52 entities, states, and 1 tribe, industry representatives, consultants, environmental organizations. So, they were asking a wide swath of different people. So they revised it. Major comments address the calculation of children's ingestion rate and duration and frequency components, which makes sense. If children are the most prone population out there, so we look at that again. So they incorporated these new, more scientifically robust children's ingestion data published in 2017, basically off of a publication, which I think we're going to get to in a few. They revised the duration and frequency recommendation. They revised the estimated toxigenic cell density based on updated values for microcystins. Okay. Incidental ingestion for age groups. Yeah, like we were talking about earlier, 6 to 10 years old, you're very prone to the effects of cyanotoxins because you're going to be in the water more often. When I was a kid, I never wanted to leave the water. You just want to stay in. You happen to swallow some, then you do. So it's based off of this publication, Dufour et al., 2006, which included 53 participants, not a huge sample size, and came up with this daily ingestion rate of about 1/3 of a liter per day incidental ingestion. And that included the 97th percentile of children's incidental ingestion, 5 to 11. And then Dufour came up with another publication in 2017, an update of that after release of the draft, and is more scientifically robust. Obviously, it's a much larger sample size of 548 participants, breaking them into, excuse me, additional age groups and recording the duration of exposure. Good ideas, right? Really good ideas. In the revised document, EPA combined two distributions. Okay, the 90th percentile of the combined distribution is the basis for the exposure parameter. So it's more restrictive. The revised ingestion rate from children 6 to 10 is reduced to less than 1/4 of a liter per day, based off of these new revisions and these new publications. They use this relative source contribution of 0.8. I'm not quite sure what that is. Deriving this value, EPA estimated that incidentally ingested water was a dominant source of exposure. Yes. So anyway, and now they're talking about this toxigenic cell density of 20,000 cells per milliliter. 20,000 cells per milliliter, which is a fairly high level. When I do algae counts, and I do them daily, I usually talk about, how many cells per liter. So 20,000 cells per milliliter is, that's a lot of algae. Okay? As a result, the concentration of cells associated with toxigenic microcystis species increased to 40,000 cells per milliliter. Okay? So the application, right? So they came up with this. I'm not sure actually what that 8 stands for. Duration 1 day, frequency not to be exceeded. Anyway, I think the big thing that I'm trying to talk about here is that, trying to get to real compliance levels or to come up with levels of cyanobacteria in the water as far as cells go, even though it's been revised and it's been far refined from its previous designation, it's still going to be difficult until we come up with some idea of why cyanobacteria produce cyanotoxins, and at least the combination of environmental variables that cause that. Okay, that's really all I wanted to talk about for that as far as HABs go. Does anybody have any questions about that so far? Anybody?
I think you're good.
All right, thanks. So we're going to go right next into, we talk about this a lot, and for good reason. It goes hand in hand kind of with what Chad was talking about earlier in, creating a population of YY males. So this time we are talking about gene drives, and we've talked about gene drives a lot in the past. And I want to kind of go through that again today. All right, the slides take a little while at first, and then eventually once they get loaded, they don't take quite so long. There we go. And we've talked about, the vulnerability of the Colorado River to aquatic invasive species overall. And it's kind of worth repeating. It is kind of the youngest large-scale engineered ecosystem in the United States. We're still not sure what's going to pop up in the Colorado River, even through quagga and even through cyanobacteria and even through all the invasive species we've seen out there. It's still a young ecosystem. We're going to get more aquatic invasive species in the Colorado River over time because those assemblages are still jockeying for position from an evolutionary standpoint. And it is, excuse me, impossible to accurately predict what assemblages may one day look like, even under, the best modeling scenarios. It's especially true under changing climatic conditions. Okay. We talked about this. Founding effects and bottlenecks do not stop aquatic invasive species that have evolved under strong selection pressures. Okay, a lot of those are going to come from places like Eurasia, especially the Orient and Asia in general. These species have been around for a long time. They have evolved into very strict selective pressures, more so than these young ecosystems like the Colorado River, which is basically at one point was kind of a clean slate because we created an ecosystem. So these things like quagga mussel explode into these relatively young ecosystems and don't like to go away, and they're incredibly successful at doing that. We talked about the genetic variability of quagga mussel in the basin. Couple different studies, both of them show that heterozygosity or genetic diversity in quagga is still relatively low, especially compared to infestations in the Great Lakes, and especially true to their native range. And that's important when we start talking about their native range. Heterozygosity is one of those things, or natural diversity is one of those things that can end a gene drive. So even if we were to engineer a gene drive in the Colorado Basin, and even if they were able to make it back to their home range, or their native range, would it wipe out this species? Well, probably not, because of that heterozygosity that exists there. However, we do expect heterozygosity to slowly increase in the Colorado River over time. They find new areas to populate, they find new areas to to inhabit, and that's one of the reasons why containment, we've talked about this a lot, and we should always talk about it. Containment is extraordinarily important. However, statistics aren't kind to even the best of containment efforts. Life wants to go other places, even despite our best efforts to contain them in an area. It just wants to go other places. So we're constantly having to work against the ecological grain of natural selection and e-migration and dispersal patterns. Right? Something as successful as quagga mussel definitely want to inhabit new areas. Okay. Go. Next slide. There we go. Spatial variability and risk assessment. Gene drives and risk assessments idealized for one area may not work in another. In other words, risk assessments and gene drives that were engineered for their home range probably wouldn't work in the first place. But certainly a gene drive or a risk assessment that was engineered for the Great Lakes or the Mississippi River drainage or any other place where quagga and zebra mussel happen to occur wouldn't work here. Right? Ecosystems all contain unique sets of variables. And there's no place where that's more true than the Colorado River. Okay? So these gene drives and risk assessments should be done on a watershed or smaller scale. Right? So ecosystem specificity in gene drives and their subsequent risks. Even though I have to say, I think that the risk involved with a gene drive in the Colorado River for quagga mussel is extraordinarily low. We have talked about this before too. Non-lethal gene drives. Now, this doesn't mean that we still don't have some degree of a sex-based gene drive, because that would have to be included as well. But non-lethal gene drives in this context doesn't mean a drive wouldn't be capable of eradicating the species. It absolutely would. Right? And we know the life history patterns of quagga mussel very, very well. Right? The development of abyssal threads, calcium uptake, we talked about that. The pros is that the non-lethal gene drives in combination with the sex gene drive, like I was saying earlier, I think in the Colorado River are relatively low risk. There's a low potential of spread of a gene drive outside of that's implemented in the Colorado River to escape and occur someplace else. Right? A gene drive would always require some degree of genetic swamping, and that's one of the things that Chad was alluding to earlier in his YY male strategies. Yeah, you have to put a lot out there. Right? We've talked about having a gene drive that's targeting the calmodulin gene, specific requirement for dissolved calcium. Right? The calcium requirement increases post-trochophore stage. And we've talked about this before too. If we could create a gene drive for something that never made it out of the trochophore phase, quagga mussel wouldn't exist anymore. It would just stop. Okay. So yeah, there's that gene drive. Calmodulin forms that critical subunit. And that's what they use to make their shells, basically. Okay. So if we could engineer a gene drive specific for calmodulin, right? The sex-based gene drives would directly affect reproductive potential in subsequent generations. Right? We'd still have to have a large number of FOD organisms, or initial organisms, to be introduced into the environment over time. Directly reproduces population through infertility. Right? We shouldn't stop there. Yes, we should have a gene drive that's based on infertility, but reproduction is one of the most highly conserved traits of organisms. Right? Life wants to persist, and it is highly susceptible to genetic variation as well. All of these are. I'm not going to say just the sex-based gene drive; they all are. But at the same time, life wants to persist. So just a sex-based gene drive and just a non-lethal based gene drive alone probably wouldn't last as long as some combination of both. We've talked all about this. I'm going to go through this real quick. Mutagenic gene reaction. Right. We've kind of gone through how gene drives work. It's a biased inheritance. If we want to talk about it again, people can send me questions, but I'm pretty sure it's pretty well known by now. One of the things I did want to talk about, the speed of a gene drive depends on its effect on individuals' fitness. So it would, as you decrease the numbers in a population, it would happen a little bit faster over time. The rate of allele conversion and on the population structure. A gene drive only requires about 30% reproductive rate for a gene drive to progress. If we had a Colorado River with 30% less quagga mussel in it, how great would that be? Okay. And that population, that 30% that was left, would be less fit than the original population. Yeah, containment. There are a lot of boots on the ground containment efforts in the basin and between among states, and that is vital because we don't want this to spread. At the same time, I don't believe that it ever would. Right? And if need be, we could have an added layer of protection via reversibility. We can reverse gene drives, basically. Talked about that. Bullen Malik's work in 2017, pretty recently. There's also this thing called daisy drives. And these are fairly interesting. These are kind of timed gene drives. And they kind of work like a multi-stage rocket. So no allele actually drives itself. They're daisy chained together. It's a built-in failsafe. So each would drive the next one in the chain, the next rocket in line. Right? The top of the daisy chain would be the payload, including the Cas9, but there's no guide RNA in this particular one. And below that would be an arbitrary number. Arbitrary, you'd have to figure out how many numbers, but an arbitrary number of daisy links in different loci, each including a guide RNA targeting the wild type version of the next higher daisy chain in the line. Okay? But since the bottom daisy in the chain has no guide RNA driving it, it's dependent on everything above it, and eventually that would be lost. So you can create these timed gene drives so that they're even safer. And I'm not going to lie, there's still a very, very large creep factor involved with gene drives. One of the things that Chad was talking about. We're also an incredibly arid region. The chances of quagga mussel escaping outside, or a gene drive for quagga mussel escaping outside of the drainage area, is low relative to other places, especially places that are more interconnected, like the Great Lakes. Potential for interbreeding with other species, it's basically zero. It's as close to zero as I can think of. The closest species out there would be California floater, and they're not going to breed. They don't. Quagga and mussel don't even, well, they do breed, but they don't produce viable offspring, and they're very closely related. So there are options for gene drives. You should be modeled. And I brought this up before, and I still think it's true. Colorado River, we do need kind of a working group for quagga genetics, because the gene drive issue isn't necessarily going to go away. And as far as I'm concerned, as far as I can think of, there is no better place to initiate a genetic strategy aimed at eradication of quagga than the Colorado Basin. There's also no better species, right? Mosquitoes are very fast-lived, very fecund. Fish are much longer-lived, right? So those gene drives take a long time. Quagga mussel are kind of the Goldilocks of species for introduction of a gene drive. They're highly fecund. You can use that fecundity against them, but they're longer lived than a lot of other invertebrates, especially things like mosquitoes. Are there other options to get rid of quagga mussel in the Colorado River? None that I can think of, honestly. I think that a large-scale eradication or even a reduction in population of quagga mussel will require some type of genetic manipulation. It'll require using their life history traits against them. Life history traits in this particular case being fertility and fecundity, but also things like we were talking about earlier, calmodulin, can't form byssal threads, those type of things. And that's really all I had on that. I know we've talked about that before, but I think it's important enough to go over again. Does anybody have any questions about that? Gene drives for quagga mussels? Did I absolutely put everybody to sleep?
Yeah, I have a question real quick, David. What's known about the quagga mussels, like sex determination, if they have sex chromosomes? I know that you and Melanie have probably talked about using some sort of YY strategy, but what information is out there as far as their sexual determination?
I'm actually not sure of sex ratios of quagga in the wild. I haven't found anything on that. I mean, they're dioecious, so there are males and females. But it's a good question. I don't necessarily know. I can tell you that the water is full of trochophores at times. Sometimes there's more trochophores in the waters of Havasu and other places than there are phytoplankton. I honestly don't know, Chad. That's a good question, though. Any other? Sorry, I didn't answer it because I honestly don't know. Any other questions about gene drives? No? Okay, we're a little over, but we're really not running too bad. Let me go over one thing real quick before everybody takes off. All right, there we go. The next thing is whether we want to continue to do this, kind of. Let's see. There it is. Future direction of this AIS task force. I mean, I certainly enjoy doing the online meetings. I, obviously prefer to do these in person. There seems to be, I was going to say, sometimes there seems to be less technical issues involved, but that's a lie because we have technical issues involved almost everywhere I go. But, I enjoy going out and meeting people, and sometimes it takes actually seeing the environment to understand it more. But we're currently out of funding. I certainly don't mind doing things like the online meetings like we're doing now. That's absolutely fine with me. Ooh, that's a horrible. What happened there? Open. There we go. That's a little better. We were funded at $25,000 a year for 3 years, and those 3 years are up. A lot of that went to travel, honestly, and website creation and maintenance, which we could still do at a nominal fee. We could, maintain or lessen the scope of services. We could have online meetings only, which I'd be glad to do for, no cost. I enjoy talking about this kind of stuff. Or we could let other, more quagga focus groups take responsibility of the task force. So I'm opening that up to questions. If anybody has any suggestions about what to do, any real input, I'd appreciate it. Anybody at all? Come on. um, like I said, I am glad to do these online meetings for free. We can talk about this at another time if we wanted to. But yeah, at some point in time, we'll have to figure out what to do with it. Like I said, I enjoy doing it. I kind of like this mix between science and application of it. That's kind of where I've always been at. So, all right. Does anybody have any other comments or questions in general? No?
Hey, Dave, this is Nick in Yuma.
Hi, Nick. How are you?
Go ahead, Nick.
You know, I think these meetings have great value for keeping people informed, and I guess if, you know, at a very minimum, people can just meet in this kind of a forum online and just exchange information, kind of like what this meeting was done. I think that's wonderful. I understand the challenges involved with no funding, but if we could figure out a way to do it, if even on maybe a less frequent scale or whatever we can do, I think, at least from my perspective, I think this is very valuable, and we'd like to try to continue to do this somehow or another.
Well, thanks, Nick. I agree. And like I said, if we just do the online meetings like this, there's really no charge. I enjoy doing these, and I enjoy keeping the website going, and I enjoy hearing more often what other people besides myself have to present. So it benefits me also, these meetings. So yeah, I am absolutely fine with that. Somebody else had a comment. I'm sorry.
Oh, this is Heidi.
Oh, hi.
Yeah, I was just going to kind of say the same thing that Nick said. And this is probably a good forum for us to talk when, you know, the agencies have opportunities for funding opportunities. So, yeah, if we can at least maintain some level of coordination, whether or not it changes due to the funding. But it's a great opportunity for all of the players to come to the table, and even if there's a need, then it can be worked out and maybe put, you know, A and C in touch with each other to work out something. So I think it's pretty valuable, and I know it kind of had some rocky starts in the beginning, but I enjoy hearing what other people are doing too.
Yeah, I enjoy that, and if nothing else, I hope we succeed in that. You know, it's always interesting to hear what other people are working on, and a lot of people don't even realize that people are working on similar lines of inquiry. I mean, we started out basically working on the Palo Verde Irrigation District Canal, looking at Salvinia, which is a relative success story, I'd say. And then we kind of expanded when Dave Britton was around, because he wanted to do that, and I did too. I thought it was a really good idea. So yeah, we expanded to include the rest of the river, because what happens in one part does have an effect on other parts. So yeah, I think this type of forum kind of pulls all that together, and that's one of the reasons I like it. So we can absolutely do this. We can just do these meetings 2 or 3 times a year. Just do them online like we're doing right now. Keep the website going, and yeah, it sounds good to me. Anybody else have any? Sorry, go ahead.
Yeah, David, this is Kurt Dusser and the rest of you all.
Hey, Kurt.
I think I've presented before on this about the Lower Colorado River cooperative weed management area that we're trying to develop and establish. We had our first meeting last March. Our next meeting will be November 28th in Yuma at the BOR office down there. I'd like to forward maybe the email to you, David, the notification about it, and if you could forward it on to this network group. But I think as far as sustainability of all these efforts, sustainability is a big issue. And we've got funding. We're going to have our third meeting. Initial meeting will be in Laughlin in January, combined with the critter meeting that the BOR leads. And the Yuma meeting, November 28th, is actually in combination with the Tamaris biocontrol workshop on the next day.
Very cool.
As the Tamaris beetle moves down in the system. But anyway, I think there might be opportunities for this AIS task force group to, and this other effort to combine or merge or have a special AIS subgroup within our cooperative weed management area if it can launch. But I think the future would be to try to get an effort like these two and try to make it happen that way with multiple funding sources to build in some sustainability. So I think that there's, to me, opportunities to try to combine with groups too that are similar efforts and similar players, so that people aren't having to go to repetitive or redundant meetings in the system. So think about that as well. And I know that that's been my kind of vision when I first got involved in this, and just basically awareness to you all. And I feel like you guys are already at the table, and we don't want to recreate another wheel. So anyway,
well, no,
some thoughts there.
I appreciate that, Kurt, and I do think that's a good idea. Invasive species don't stop at the water's edge, so there is that, right? And invasive species biology in general it's a little different between the aquatic world and the terrestrial world, but they're close enough to be at least, sister groups. Does that make sense? Absolutely. I mean, having everything under one comprehensive roof probably isn't a bad idea. And, there's a lot of groups focused on quagga, so I try not to be repetitive in that. We've talked about a lot of other things besides quagga. But quagga is always going to be the 800-pound gorilla in the room, and there's no way around that, and rightfully so. But yes, I do agree. If we're going to cover all invasive species, then we should have an aquatic end of it, we should have a terrestrial end of it, or a semi-terrestrial end of it. And like I said, sister groups along those lines sounds like a good idea to me. Does that make sense?
Yeah, it does to me, I think.
Thank you, Kurt. Any other comments? Any questions? All right.
I think my concern is just getting this CWMA launched, and if it becomes sustainable. And I think you guys are already, or this group is already, ahead in the game, but it's again running into sustainability. So I think Absolutely. If you can way to combine, you can capitalize on that.
No, I absolutely agree. Yeah, no, there are joint messages in both, right? Invasive species are invasive species. So yeah, I absolutely agree. Mm-hmm. Anybody else have any questions, comments? Okay, then I guess what we'll do for now is we'll probably have these online meetings every, 4 months or so. It's been a while since our last one, and I realize that. But if we can have these every 4 to 6 months, does anybody have any recommendations about timing? 4 or 6 months we have these meetings? Daily? All right. Then what we'll do is we'll shoot.
Right, daily it is. I heard daily.
I lied, Kurt. I lied. I think shooting for 4 times or 3 times a year, every 4 months is doable, because we don't want to be repetitive. We want to talk about new things, right? It is a good way to get people together to talk about those newer things, whether it's science, whether it's management, whether it's the science of management. Then this is a good forum to do that in, I think. And I certainly enjoy it. So it teaches me a lot. All right, then that is the plan. It was a good group today. talked about a lot of interesting stuff. And I'll send out a reminder for the next meeting. What is it? September, October, November, December, probably sometime right after the holidays. Okay. And we'll have another meeting like this. And if anybody has anything that they'd like to present, please let me know.
Okay.
All right. Thank you very much. I appreciate it.
Thank you.
Thanks a lot.
All right, thanks everybody.